Hello, hello. How's everybody doing tonight? Good. Okay, I sound a little bit sleepy. I feel very awake. I'm caffeinated. I hope you all are caffeinated. My name is Sarah Grummett, and I'll be hosting Cafe Sci this evening for you. Thank you so much for joining us. Cafe Sci is presented by PPG. It's also supported by good folks like yourselves. We want to thank everybody who has donated to support this program. If you're unable to donate but would still like to support Cafe Sci, the most helpful thing you can do is to invite your friends to join us here in the audience. Cafe Sci takes place on the first Monday of each month, and you can register for next month's speaker at carnegiesciencecenter.org. Before I go ahead and introduce our speaker for the evening, I'd just like to remind everybody that there will be a Q&A after the talk. For our on-site audience, most of you, I think, know how it goes by now. You can ask questions by raising your hand, and we will bring you a physical microphone. Please wait until you have the microphone to ask your question. It makes it easier for both our presenter to hear you and for our audience at home to hear you. For our online audiences, you can ask your questions by using the Q&A button at the bottom of your Zoom window. Online audiences, you can enter your questions at any time during the presentation, though we will be holding all questions till the end. And it's my pleasure to introduce tonight's speaker, Lance Davidson. Let's give Lance a round of applause. Lance will be presenting on Gears of Life, the mechanics of how we are formed. Lance is an American bioengineer and developmental biologist at the University of Pittsburgh, where he is a William Kepler Whitford Professor of Bioengineering and also acts as the director of Mech Morpho Laboratory. He received his bachelor's degree in physics from the University of Illinois, a master's degree in space science from York University in Toronto, and a PhD in biophysics from the University of California at Berkeley, with a thesis titled The Biomechanics of Sea Urchin Primary Invagination. His postdoctoral training was in cell and developmental biology at the University of Virginia, where he transitioned to working on frog development. Davidson established his lab at the University of Pittsburgh in 2006. From his PhD thesis work through the present, his research interests include the physics of embryonic development, the genetic and mechanical origins of birth defects, and the role of the environment in embryonic robustness. Now let's give another big round of applause for Lance Davidson.
Is that working? Yep. Well, thank you, Sarah, and the Carnegie Science Center for inviting me to give this talk. And this is particularly, I have a deep love of public science lectures. As a graduate student, I had the chance to attend Friday evening lectures at Woods Hole at the Marine Biological Labs, and those were just so wonderful to attend, and I'm trying to get a little bit of that feeling here tonight. So what I'm gonna, so this is of course a title slide. Is there a way to get the lights off on this, on the front a little bit? Okay, all right. So first off, what I'm gonna talk about is really try to guide you through, and maybe not, what was that? I don't know. Nobody saw. But one of the things that we're very interested in is how genetics controls the formation of the body. And in particular, these are just this is a short time lapse of pictures of individual, yes, perfect, thank you. A time lapse sequence of individual cells. So a cell is about this big, and these are fluorescently labeled molecules, proteins within those cells that are showing you in magenta one of the cytoskeletal members. So the skeleton inside of a cell, and one of the motors on that skeleton here in green. So you can see this is a very dynamic process. Cells are moving around constantly. They coordinate their movements to generate force production and to move tissues over time. And so I'm not going to come back to this movie, but really this is where we start. We think about how molecules create forces in the embryo. So I do have a brief video that I want to show you first before I talk about frogs and morphogenesis.
Sometimes scientific breakthroughs come from the strangest sources. For example, frogs. But what can we learn about them? What can we learn from them? And what can they teach us about ourselves? It turns out quite a lot. Since 1993, Cold Spring Harbor Laboratory has hosted a course called Cell and Developmental Biology of Xenopus, dedicated to the study of the African clawed frog. You see, these frogs have several unique traits that give scientists a leg up in their research. One is being see-through. Another is quickly developing from an egg to an adult. They help us understand how organs work in all types of life on Earth, from single cells to complex organisms. Cloning, stem cells, organoids, genome editing, all have froggy origins. Every year, students from around the globe make the jump to CSHL to learn the basics of this extraordinary amphibian. They learn to use state-of-the-art techniques and equipment such as live imaging, bioluminescence, and microsurgery. Here, the blood flow and heart activity of a tadpole are rendered with stunning clarity. A brain, an eye, and the surrounding nerves are all imaged with great detail. Since its inception, course alumni have formed a supportive and tight-knit community with members worldwide passing on their knowledge to others in the field. It's a glowing example of why CSHL's Meetings and Courses program remains a hopping hub for cutting-edge science education.
I just wanted that little blurb there to introduce you to the model organism that I work on. And so next thing I want to do is introduce or thank actually the people who did all of this work. These are my group members at the University of Pittsburgh, in particular my 4 graduate students, Jing Yang, Geneva Masek, Summer Anjum, and Yicheng Dong. Are way back there. They hang out at the back all the time. And I just wanted to thank them and our funders. The research that we do is made possible by funds from the NIH, the National Institute of Child Health and Human Development, the National Institute of General Medical Sciences, the National Institute of National Heart, Lung, and Blood Institute. We've also had funds from the National Science Foundations, and also we have now some funding from the Navy through the Office of Naval Research. So I want to have a sort of outline. I'm going to first talk about who I am. We have a little bit of that already. I'm going to tell you a little bit about development and why it's such a big mystery in biology still. And I'm going to follow that up by saying, leading you through, which the video already did, why frogs are such a great way to explore this mystery, and then how we in the lab use physics and engineering to shine light on this particular mystery of morphogenesis. And so for me, I was born in Chicago, so I'm a Midwesterner at heart. I went to undergraduate at the University of Illinois in Urbana-Champaign. And I went off, and so I was a physicist as an undergraduate by training. Lots of math, no biology. I didn't take a biology course as an undergraduate. I really liked in physics where physics rubs up against other fields. Geophysics, atmospheric physics, space science, astrophysics. And so I went and did a master's degree in space science at York University in Toronto. And then I went into industry. And in this case, this is the military industrial complex at the time. We were doing work for the Navy at Fleet Numerical in Monterey, California. And so at that time, I was sort of living the dream that many undergraduates have, which is, you spend all this time in undergraduate, you work hard, you burn out a little bit, and you wish that you had one of those 40-hour-a-week jobs where Friday at 5, you go home, you don't have to work on homework or problem sets or anything over the weekend, and then you start up again on Monday with the work week. And I had that life. I thought it was, but I filled every part of that, of my time off doing science hobbies. I love to go explore the tide pools on the California and Oregon coasts, hiking along the coastlands. And so I read voraciously, just reading anything that came into my hands. And at one point, I read this book by James Gleick titled Chaos. And this was the late 80s. And this book really sort of clarified for me what I was missing from my life, right? So we think now about work-life balance, and I had a gap in my life. I wanted to continue doing work, and the kind of science that was described in this book was really appealing. To me, it was the physicists and mathematicians and engineers were coming to the aid of biology and biological questions. And so, so I took a turn. I took a turn from where I was working, and I re-entered graduate school in a PhD program at UC Berkeley. I fell in with a particularly shady group of characters. My graduate advisor, I had 2 of them. How much luck could you have? And they brought me in, and I learned about biology and developmental biology as an adult. I feel very fortunate to have had that experience. And from there, I went on to do a postdoctoral fellowship at the University of Virginia, where I took what I had as a solid foundation in physics and biophysics and added to it what I call wet lab biology, molecular biology, cell and developmental biology. And then from there, I joined the University of Pittsburgh faculty in bioengineering in 2006. And so I've been here almost 20 years. So how does this strike me about how biology has come to investigate developmental biology? So I think about this historically. So in the late 1800s, biologists were interested in cataloging and characterizing everything in nature. And these are just some beautiful paintings or drawings by Ernst Haeckel in 1904 called Kunstformen der Natur. This is the forms of life, basically. And so these many panels just beautifully illustrated. These are jellyfish, cnidarians. Over here are echinoderms and sea urchins of various sorts. And then in the middle panel, this is my favorite right now, these are frogs. And so as you can see, this was the sort of characterization, this was what biologists were looking for at around the turn of the century. Now, there were other things happening in science, and at that time, it was also the birth of modern physics and chemistry, the birth of what I think of as elastic mechanics, engineering, and these sort of terms that I'm going to introduce you to were actually getting very stabilized and characterized mathematically, very sophisticated mathematics at the end of that century. So everything that we think about in terms of stress and strain and modulus and springs and dashpots, all of that was worked out by the late 1800s. This, and in chemistry, the same situation, electricity and magnetism. Quantum mechanics was coming online just sort of shortly after this. And so there was almost like a rebellion amongst biologists where there were, this is Wilhelm His, an early embryologist in 1888, to think that heredity will build organic beings without mechanical means is a piece of unscientific mysticism. Wilhelm Roux said, for biology to be a true science, it must adopt quantitative and mechanical methods, i.e. that it must model itself after the physical sciences. And for the biologists at that time, and for these guys, this was like fighting words. They were just like, you know, they were just ready to wrestle or box each other or something. I mean, this was pretty heated debates going on. And so as I continue, I want to get a couple of terms defined, and so that, you know, I'm going to try not to use so much jargon, but there's a couple of terms that I really have to catch you up on. And I think the last month's speaker defined biomechanics very well. And so that's the study of mechanical laws responsible for the movement and structure of living organisms. We have a certain biomechanics, and that biomechanics goes from the principles of walking and gait all the way down to the principles of the molecules inside of cells. So biomechanics is really tremendous. It doesn't matter what scale you're looking at, the principles of biomechanics still are in place. It's the physics of biological mechanics. But on the flip side, there's this thing called mechanobiology, and it's a kind of funny word. It's the exact opposite of biomechanics. You put one word in front of the other, or you switch places, and it means almost the opposite. It's not quite the opposite, but mechanobiology is the study of biological mechanisms used by cells and tissues to sense and respond to mechanical signals or cues. So this is the biology part, actually taking a reading or sensing the mechanics, the biomechanics, and changing the biology in response. So these are two aspects. I'm going to talk mainly about this upper one, the biomechanics, but there is a really important role that the mechanobiology plays. And so when I talk about developmental biology and organisms, how they develop, developmental biology is just the study of processes that drive growth and development in plants and animals. I'm only going to talk about animals here, but the same principles work in plants. I have many colleagues that study plant development and mechanics, and they have a much easier than we do. Their cells don't move around like ours do. So, you know, I think of it as an easier situation, but they would debate that. And then there's this term morphogenesis, which actually is it's embedded in the name of my research group, mechanics of morphogenesis. And morphogenesis is the shaping of an organism or their organs through differentiation of cells, tissues, and organs. And so that's a big part of what we do is understanding how morphogenesis takes place. And so there's two particular types of morphogenesis, two events in morphogenesis in early animal development. The very first one takes a ball of cells early in development that are pretty much indistinguishable from one another and rearranges those cells into three different layers. The outer layer of the embryo is going to be the ectoderm. And that's the side of the embryo that's on our outside right now. It's the part of the embryo that faces the outside world. And then there's a group of cells that are on the inside, the endoderm, and that's going to eventually form the lining of your gut and your lungs. And then there's the middle part, the mesoderm, and that's going to give rise to your muscles and bones. In early gastrulation is the process of sliding all of those cells into these three different layers. And it's a common process in all animals. Gastrulation is a massive rearrangement, a repacking of cells in the embryo. It is common that they have to rearrange to accomplish this. There is no organism that just sort of magically kind of teleports these cells into place in their final location. They all have to move. And then lastly, a feature of our own development that differentiates us from many other animals is the process of neurulation. This is the process that happens in vertebrates that gives rise to the brain and the central nervous system. This is one of the last large movements in the early embryo. And it establishes the brain, the the spinal cord, and the central nervous system through a process of folding tissues, okay? And so I love this quote. This is one of my heroes in developmental biology, Louis Wolpert. He came to the field from, he was a South African, and he came to the field from civil engineering. And in South Africa, he did his degrees in civil engineering, and then he moved to London to get his graduate work, and he was like palling around London, apparently, as South Africans would do, and he was bored with the options of becoming a civil engineer, and hung out with, again, he found some disreputable people in London, and they introduced him to studying embryos. And so I think this is, gastrulation, which is the most important time of your life, yes, but I have a little addendum to that, which is that without neurulation, well, you wouldn't appreciate the importance of gastrulation, right? So this is something that Wolpert, I think he would appreciate that addition. So I'm particularly interested in the question of how morphogenesis works. And when you think about work, you think about pay and 40 hours of work a week. We'll get to the difference between that, but I want to know how the embryo assembles itself and what pushes on what and what kind of forces are at play. And then what makes morphogenesis so robust? So it may seem that it's so fragile. Embryos are so fragile. When we in the lab fertilize a batch of embryos, if they make it out of, if they fertilize, that's good, that's an important thing. But if they fertilize and start to develop, we can get hundreds of embryos developing perfectly normally, sitting right next to one another, and they're in sync. And so what makes morphogenesis so robust? And what do I mean by that? So here are two embryos up here. This is a normal frog embryo developing, and it's in the process of shaping its neural plate. This is where the head's gonna be, this is where the tail's gonna be, and it's taking this sheet of cells and folding it in, and that's gonna be where the spinal cord is. On the right-hand side, this is an embryo that has been injected with a protein that will disrupt that. Okay, so this protein is injected and expressed in each and every one of these cells, and it clearly, you don't need a PhD to recognize that the one is different from the other. This one has a little sphincter right here that closes up in the middle, and its head literally never gets away from its butt, okay? It's very important for the head of the embryo here to get away from the butt of the embryo here. It's pretty simple. These embryos are doomed. And the reason for that is not just on the large scale, but on the very finest scale. The cells inside of this embryo are moving in such a way that they exert forces and they fold this tissue in this direction. They basically adopt a behavior that's very stereotypic. The cells extend out to their neighbors and they go sort of like this and pull on their neighbors. Cells that are expressing this mutant protein here are just dumbfounded. They're lost. They don't know one way from another. And so they're really not able to generate the sort of coordinated movements in the tissue as are needed. Okay. So this raises the question of why does morphogenesis fail? And so normally in development, I told you, like the embryo is so robust, we can do things to embryo development and try to perturb it and it recovers. But in many cases, the embryo will fail. So I sort of go back to this question about mechanics and that early work of these sort of fighting Germans, Wilhelm Roux and those, and they inspired a whole several decades of research in Europe, and Darcy Thompson summarized that in this beautiful book on growth and form. And so he sort of pulls in all of these ideas about the roles of mechanics in shaping organisms, responsible for the evolution of organismal shape and size. And you may have seen like these pictures of fish that are drawn on grids, and you just sort of stretch that grid of a fish, and it goes from being a sunfish into a trout or something like that. And this book is full of physics, and it's full of biology, and it's just a beautiful example of the sort of thought of how mechanics could actually control the shape of an organism. And so this sort of outlines the kind of way that we think about mechanics in the embryo, and the role of mechanics, biomechanics in the embryo. And so at the very top of this hierarchy, we have this thing called the genotype, and that's the proteins, the genes that are in that embryo, the signal transduction pathways, all of these important features of how cells talk to one another, how cells maintain their skeleton, how they assemble things outside of the cell. And so this is the genotype. This is the thing that we inherit from our parents. And that genotype then produces proteins that make cells. And these are cells that I was just showing you, the cells that rearrange and the cells that make cytoskeleton, and that those cells coordinate their movements, and they're responsible for this tissue mechanics on a larger scale that folds things and pushes layers of cells between one another. And at the end of this hierarchy, we have the phenotype, whether you're a human or whether you're a frog. This is the pathway for robust development. And so Darcy Thompson would be just like happy with this hierarchy. Of course, he didn't know about genes at the time, or he wasn't really thinking about genes at the time. He did know about them, but he didn't really think too much about them. But there's this other thing that we've been seeing more recently, which is that tissue mechanics can feed back and start to influence how cells operate, and how the genotype can even be subtly changed. And so I think Darcy would be like, huh? What's going on? And then the fact is that these feedback mechanisms actually give rise to robust development. They're the thing that's responsible for robust development. And so a confetti brain, right? I mean, of course, this is Darcy Thompson's response, because all of this down here is completely new to the kind of end of the century perspective of the role of mechanics and biomechanics. This is the contribution of mechanobiology. Okay, so when I talk about mechanics, I'm throwing the word around like really loosely. Like, what does mechanics mean? Is this like, hello, my car won't run, I need a mechanic, right? And so we have these really interesting ways of talking about mechanics. And so I like to have this section in my talk called disambiguation. It's one of my favorite pages in Wikipedia, where you have a word that can mean all these different things. And in English, disambiguation is important because we have all of these terms that we use. We frequently use movement, and this is the definition of movement. You take something, and you lift it up, and you walk it over here, and you put it down, and that's motion, movement. Now, strain is something else. When we talk about strain, often like it's the strain of life, it's the strain of some, tomato that you're gonna grow, or something like that. There's so many different ways in English to use this word. For us, in engineering terms, strain is really a single measurement of the length of an object before it gets deformed, and then the length of an object after it gets deformed. And so we can calculate strain. This is not something you hope that happens to yourself, but in terms of strain, we can calculate that. And then when we talk about force and stress, oh my gosh, we are forced to do something by, some rule or law, or you're under stress at the workplace. Well, stress has an actual quantitative definition here as the ratio of the force applied to an object divided by the area or surface area that it's applied to. So if I go and push on a wall, the force that I'm exerting with my hands will be against a surface, an area that my hands cover. And so that's going to result in stress, okay? And then lastly, mechanical properties. These are things that we're sort of intuitively familiar with, right? So we think about viscous materials. Viscous materials, I hope you guys enjoyed some of this before coming tonight. Of course, this is apple juice. But what happens with fluids is that you apply a stress to a fluid, and it moves out of the way and comes immediately to rest, okay? And that's really a definition of viscous behavior. Now, we're all familiar with elastic objects, where you deform an object, pull on a rubber band, and you let go, and it goes back to its original shape. And then there's something called viscoelastic materials. Now we're getting into things, sort of material properties that are very much more like biological materials, or at least materials in the early embryo. And memory foam is a great example, right? I could sell you a memory foam mattress because they're so wonderful. But when you put your hand down onto memory foam, you apply stress, and then you pull it away, there's a time that you can still see the imprint of your hand. And that's an indication that there's that the material has been deformed, and it then has to relax to adopt its shape. So you come back 5 minutes later and you can't find your handprint anymore. And that's a viscoelastic material. And lastly, the materials that we typically think we see in embryos, that we measure in embryos, is something more like a viscoelastic plastic. And that is if you had a memory foam mattress that was viscoelastic plastic, your handprint would stay there forever, okay? And this is an example. I don't know if you've ever encountered a material like this. Silly putty, exactly. So silly putty is great. You can bounce it, it's like an elastic, and then you pull it out like taffy, and it's plastic, and then you can roll it back up, and it's back to being elastic. Okay, so this is something that we think of as more about what real embryos are made of. Okay, I'm going to take you back to frog for a second, but just after I sort of explain why we pick frog for our studies. And so there's Krog's principle from the early 1900s. For a large number of problems, there will be some animal of choice or a few such animals on which it can be most conveniently studied. This is sort of like, yes, of course, right? If you want to study the genetics of plant development, you don't pick giant sequoias, right? It's going to be 2,000 years before you get the next generation. We don't have the time for that, right? So, so when we think about model systems for development of vertebrate animals like ourselves, we think of really these sort of three different organisms. One is the Xenopus frog, another is the chicken, and then there's mouse. You don't want these in your home, but some of you have probably even had a zebrafish in your home. You pick any one of these, you can study developmental biology in them. In general, they're models for developmental biology. And so these are 4 time-lapse movies of these different or at least 3 time-lapse movies of these different organisms. These 3 develop outside of the mother. So they're very useful for following things moving and for time-lapse studies and dynamics. The one that's closest to us, mouse, is of course develops inside the mother, which makes it very difficult to study what's happening live. And once you take a mouse embryo out, it only has a few hours in which it's viable and continues to develop as if it was still in the mother. By contrast, you can take a frog embryo out of the dish, and actually I have in my bag, I've got some frog embryos with me. that are developing right now. I mean, they're actually developing fine in my bag. And if you want, after lecture, come down, I'll give you they're very tiny. You can, but you can take a look. And so we focus on Xenopus laevis as a model system because it is relatively close to man, and it might not have been a point that was appreciated in the 1800s how close it was to man, but in the recent 30 years or so, genomic analysis has indicated just how similar all of these models are to man. Most of the proteins are identical or close enough. They can be interchanged. The genomes are so similar enough that one gene next to another gene in human is next to the same gene in frog. So even the orders of genes on chromosomes is the same from all these species. So the frog embryo has been the source of, or amphibian embryos have been the source of Nobel Prizes. This was the very first Nobel Prize made possible by the ability to cut a piece of the embryo out and microsurgically move it into another embryo. And this experiment was just crazy. What they did was they cut a piece of an early embryo out of a donor and moved it over and put it into a host embryo that was at the same stage, and they could grow them up. And lo and behold, that little piece of tissue created a second entire head and axis in the host embryo. So this is a fantastic discovery that led us onto many different paths. So this is one of those things that was a serendipitous discovery, although I don't know what the Germans were doing, grafting pieces of embryos around in the dish, but they did this, and this was the very first Nobel Prize that was won. Frogs continue to have important roles in modern biological research and biosciences and biomedical research. Here's the 2012 Nobel Prize in Physiology or Medicine. On the right-hand side is Shinya Yamanaka. Shinya Yamanaka developed the induced pluripotent stem cells, or iPSCs, which are very important subjects for modern research. And John Gurdon showed that very principle that you could take and reprogram a cell into an embryonic state. And so John Gurdon did this work in frogs way before Shinya Yamanaka did. And what he did was he took, this is his colony of frogs, he was quite a bit younger here. This was in the 1960s. And he took nuclei from one frog and injected those nuclei into the enucleated egg. Now this was an egg that didn't have a nucleus, that had been taken out. And if you place that nucleus into the egg, the embryo would develop from that. And he could take lots and lots of nuclei, not just from another egg, but from a fully differentiated skin cell. So this was a really amazing thing, to take a cell that was already differentiated in an adult animal, take it out and put it into an embryo and see it reactivated. And so he had done this with albinos, albino frog skin, and put it into pigmented eggs. And these are all the clones, basically, of that albino skin cell, skin tissue. And so much later, this was applied to create Dolly the sheep. Everybody knows who Dolly was. And then there's also those of you who don't have much head for science but watch a lot of television. It's also the source of the fictional clones of Tatiana Maslany's character in Orphan Black, right? So same process, somatic nuclear transfer. All right, another great advantage that we have in frog is that we can take an early embryo and inject into a single cell whatever we want. The embryos are available to us, they're fertilized, we have hundreds of them in a dish. And then we can, what we see from those embryos is we can track what cells are doing with these fluorescent markers. And so I'm not going to talk about microscopy, but at the same time that, all of these techniques have been developing, new techniques in microscopy have been coming on board. And so we can do the same thing. We can even look at different sides of a tissue. So here's one side of the skin. This is the side that faces the outer part of the world. And then this is a basolateral position. This faces the inside of the embryo. And I think you can even tell the difference between the behavior of these proteins in these different locations. On the skin side, it's very calm, very peaceful. It's very rugged. It's there to prevent, disruption by the outside world. And on the inside, this is where things get moved around. This is where the cells are pulling on each other and rearranging. So this is sort of an advantage of working with frog. So, okay. If you work on frogs, you find that frequently people send you pictures and bring you things from every gift shop in the world. You can be in Paris, and there's, Eiffel Towers, and then there's frogs. And you could be in New York City, and there's the Statue of Liberty, and then there's little frogs. So I can guarantee that frogs are like the most wonderful things in the world. So, all right, we're doing fine. All right. So this is a time lapse. This is what this frog embryo looks like when it's developing. And this is about 13 hours of elapsed time. And 13 hours is just perfect for the professional scientist. It's from the start of your day to the end of your day. You can finish up work, tidy things up, and then go home and start again tomorrow on this. And so what we see here on the left-hand side is sort of the life cycle of the frog embryo. And so from the egg to go through cell divisions, it's about 8 or 9 hours. It begins this process of gastrulation and neurulation. By 24 hours, the embryo is basically, looks like us. All the parts are there in the right places. The hearts are going to form about a day and a half later. The central nervous system is forming. All the organs form well before, like sort of right around this time. And then it's another couple of months before maybe a year, before they're ready to lay eggs again. And so the frog embryo has a tremendous number of advantages to us. One of the more important parts is this one right here, microsurgery and dissociation. It's really like a miracle. Not to say not a miracle. It is a phenomenon that is amazing, is that you can take a piece of this embryo, cut it out of the embryo, take that piece of tissue and put it in a dish and put it someplace else. It will continue to develop as if it was still in the embryo. Programs of development turn on, cells communicate with other parts of the tissue, they build things, they undergo morphogenesis in isolation from the rest of the animal. And so that gives us a very, very powerful tool to investigate the process in isolation, separate from what's going on in the tail or what's going on in the head. And so by isolating that, we can really dig out what's happening. And this is just giving you an example of, just how simple and straightforward some of these things are. I know doesn't seem so straightforward. This is the best thing ever. This is a dish of fertilized embryos that are, you can see they're pigmented on one side and they're white on the other. When they're like roly-polies, right? Where you push them over, you can see the white underbelly, but then when they roll back, you can see the pigment on the top. And they can be injected. This is an undergraduate in the lab a number of years ago who could inject embryos. These are only 1 millimeter across. You could inject them freehand with a needle. So, young people these days, right? And then over here on the right, this is a microsurgical manipulation. This is my forte of peeling layers off of an embryo. Now remember, this is 1 millimeter across, and I can peel, it's like peeling a grape, but imagine that the grape is only 1 millimeter across. And so I'm a microsurgeon. That's what I did in Virginia, and that's the kind of thing that we do in my lab. So I don't have many equations. I have two, okay? I'll come clean on it with you. I have two equations. This is the one to pay attention to. It's fairly simple. This is deformation. This is when we talk about mechanics of the embryo. This is when a piece of tissue moves from one time point to another, changes shape. And that shape change is driven by forces. Of course, we know about forces. You have to push on something to get it to change shape. And then it's divided by the mechanical resistance of that material. So if you push on something that's very stiff, that has a lot of mechanical resistance, it won't change shape. If you push a little bit on something like Jell-O that's very soft, that offers little resistance, you'll get a lot of shape change. So we can understand how mechanics works inside of embryos with just this simple equation, okay? Now the challenges are that deformation, like those movies I was showing you, are things we can watch. We can just turn a microscope on, we can set up a time lapse, we can look at how these things are moving. And this is great. We don't have to perturb embryos at all. This is just, my engineering friends like to make fun of developmental biologists. They say, oh, they just like to watch. Okay, all right, okay, I get it. But on the other hand, if we want to go and look at force and mechanical resistance, these are things that we have to measure. We have to pull that piece of tissue out. We have to subject it to a known force and see how much it strains. We really have to perturb cells and tissues to measure this. Now, when we take this idea of measuring these properties of embryonic tissues, we have a problem. That problem is major technical challenges to measure force, stress, and stiffness. They're tiny, right? So I showed you an embryo. It's a millimeter to start with, and you start cutting it into pieces. Now you're talking about a piece of tissue that's 100 microns on a side. If you have hair, then you pull a piece of hair, and it's 100 microns thick. So that's a thickness of a human hair. So this is very tiny pieces of tissue. They're ultra soft in how they deform, so their mechanical resistance is very, very low. There are not that many places on the human body that you could go to that have such low mechanical resistance. I'm pointing to one right now. But most of the rest of us, our bones, our muscles, are thousands of times stiffer than that. They're super viscous, and by viscous, it's like, think about peanut butter. They're more viscous than peanut butter. So it's a different kind of material property. And then ultimately, these tissues are active in that as you're like trying to make mechanical measurements as something that is inert, it's pushing back. It's generating forces on its own. And it's generating forces in the scale of 1 to 2 micronewtons of force. Now, I know you probably don't have an intuition for what is a micronewton, but I'll give you an idea. If you take a fruit fly, which is one of our competitor model organisms, and you dry it down, you desiccate it, and then you put it on a scale, the weight of that dry fruit fly is going to be about 2.5 micronewtons. So very, very small forces. Okay, so I don't want to go into like knock you over, but in the 20 years that I've been at Pitt, my group has had biophysicists and engineers and mathematicians and physicists and biologists, and we've built a whole range of devices. And I think I really take pride in the fact that we've developed new tools to measure these sort of very fine things. But we love to have fun in the lab, and we name them in the lab we call them these things. We have other names that we use for granting agencies. But there's the histo-wiggler. I'll give you an example. This is the nanonewton force measurement device. But in the lab, it's the histo-wiggler. That's a long phrase. We have the embryo sucker, where we can aspirate a tissue from a small piece of with a pressure. We have an embryo poker, where you can stick a little microprobe in and like a finger in a pie. We now have a tissue tractor in the lab. One of my graduate students, Jing Yang, we have a patent pending for this, and we can apply extreme strains. Like you start like this and you pull it out like this, and that's a huge amount of strain. And then we have another device that's under development now. This is a collaboration with some folks at Carnegie Mellon University in mechanical engineering and robotics called the handheld tissue force microscope, so that we could take a little probe and just sort of move it around on the surface of an embryo and measure its mechanical properties. We've got to come up with a good name there, but this one's too long. So what are some of the lessons that we've learned by applying these tools? The first one, I think, and we were talking about, say, this role of cytoskeleton, this skeleton inside the cells. It is one of the major sources of mechanical properties, both the material properties as well as the forces that are produced and the mechanical resistance. The embryos at this stage don't have bone. They don't have tendon in them. They're just cells stuck together. And so this kind of dynamic process inside of an individual cell, here you can see how it's organized in a cell that's been cleared with a microscope. And so embryos and organ primordia are ultra soft when they form. And this is turning out to be true across all different model organisms. Organ primordia assemble from multiple germ layers. And this is when I was telling you about the process of gastrulation, making these sort of layers of cells within the embryo. These are where your liver comes from, and your heart, and your kidneys. They come from associations between multiple germ layers in the embryo. And then lastly, organs form really, really early in the embryo. So when we think about this, like going from an embryo egg to an organism, it's not the heart and all of these major systems of physiology don't start up here. They start up way, way back here. So in the frog embryo, the first beating heart forms out of a mass of cells, and I say mass, it's about 100, maybe 50 to 100 microns in diameter. A very, very tiny little pump that's going to keep on pumping throughout the life of the frog embryo, all the way through the larva, all the way through the adult. And over that time, that heart grows and remodels and changes its function, goes from one fluid regime to another fluid regime. And it's just pretty remarkable, and all through this time. So organs form really early, and they form really small. And then from what we've found, and it's common in most other organisms, is that altered stiffness, and so there's a change in embryon of the stiffness of the tissues over time, but altered stiffness induces developmental defects. So if you make those tissues too stiff, you get a developmental defect. The embryo doesn't gastrulate normally. It doesn't neurulate properly. It won't make a central nervous system. Often we find, though, that if we soften embryos, they're perfectly fine, perfectly happy. We're still trying to understand why is it that if you soften them, you take them off of this path of mechanics, you soften them, they're fine. You stiffen them, it's grim. That's not really a good end. Okay, so this part I want to the last part is I want to tell you about how we take these tools and our way of thinking about science and test something. Okay, so one of the things that we discovered was that as embryos age, they get stiffer, and they don't get just a little bit stiffer. This doesn't really show the drama exactly. This is about an 8-hour period in time when the embryo increases in stiffness by 6 to 8-fold. It's like you woke up in the morning and you ran up these stairs, and then 8 hours later, you are like maybe 6 or 7 times heavier, and you aren't running upstairs anymore, right? So the changes that are going on in the early embryo are phenomenal, and they're happening just as we're watching them. So those time-lapse movies, they're getting stiffer and stiffer and stiffer, but we can't see like any change in how the embryo's developing. So we want to know how do these structures at different scales contribute to this elastic modulus? It goes back to the kind of questions that we are interested in. And so we have these kind of questions all the time. And so part of my group's experimental, part of our method is to adopt an experimental design that follows this method called strong inference. And this is where the bulk of great discoveries have been made in the 20th century in science. And the basics of strong inference is that you can never prove a hypothesis. You can never prove it. You can keep coming up with more support and more support and more support. It could still be wrong. And this is where science policy and science communications often get scientists in trouble, because if they believe this for truth, this is a hard thing to communicate. You can never prove a hypothesis. So what you want to do is you want to start your project with multiple hypotheses and then design experiments intentionally to disprove your hypotheses, okay? So this is a really hard concept. This may be different from what you learned in elementary school about the scientific method, but this is how we try to operate in the lab. And then lastly, we try to only carry out experiments on hypotheses that can be falsified. If you have a hypothesis that can't be falsified, how do you disprove it? So those hypotheses we have to discard, and we have to redefine a way that we can falsify them, that we can disprove them experimentally. Okay, so sort of sum it up in a slightly different way, all right? So I think, Sherlock Holmes here, of course, Benedict Cumberbatch, when you have eliminated all which is impossible, then whatever remains, however improbable, must be the truth. And that, I think, actually does summarize strong inference methods quite well. And so here we go. We try to do something like we're going to alter the genotype, and then we're going to look later and try to observe how tissue mechanics has changed. This is the basis of many of our experiments that we do. All right, now this is the, as I say, in the lab we have a lot of fun, so this is like one of the things that you discover on the internet, graduate students reporting all of these various possible ways in which experiments that are planned go horribly, horribly wrong. And yes, there's tried, failed, ran, repeated, also failed, worked. This one here is my favorite, caught fire. If you're not working hard enough, your lab isn't catching fire, okay? I do believe that. And then there's these experiments that are impossible, and they hopefully get stopped pretty quickly. Of course, most of the experiments are too expensive, and only some of those are really worth publishing, of course, right? So we started off with a set of structures that we thought might be important to the mechanical parts of the embryo, to define that mechanical part. And so we have them going from one scale up here, from tissue architecture, where we think about the large scale structures in the embryo, and then it goes all the way down here to those molecular factors that I showed you in the very first slide. And so, okay, so we have this idea, and all right, we have our plan here, the genotype. We want to alter specific factors, and then we have our observations. We can use microscopy to confirm that that these changes did what we thought they were doing, and then we can actually measure this modulus, this mechanical resistance at the end. All right, so this is the ideal goal of those kinds of experiments, but we have to have something else. we have to have a model that will relate how a specific factor changes the elastic modulus. And so when we think about these different structures at different scales, this requires us to have some supporting model that we're going to use to make predictions and then try to falsify those predictions. And so we were so lucky when we started to think about this that there was a beautiful theory developed by Lorna Gibson and Michael Ashby. Lorna Gibson at MIT and Michael Ashby at University of Cambridge. And they had been studying things called foams. And on the left-hand side, you can see these are classic synthetic foams. And this is like what you get from Amazon in the package, or wrapped up, boxes of things that you get shipped to you. This is what it looks like on a fine scale. And they also noticed that many of these synthetic foams have a very similar structure to these biological foams. And this down here is over here is bone. This is actually in a plant. And these are all sorts of different biological, naturally occurring foams. And so they developed a theory that would relate all of the mechanical structures that we were interested in into elasticity. And to do that, this is like a joke among physicists, imagine a spherical cow. That's the punchline, I'm sorry. But they imagined that the foam was defined by a single unit cube. And you could make all these measurements, you have all the knowledge of the materials. This is a closed cell foam, like our cells. And they developed this very nice, this is the second equation that I was going to show you. You don't have to worry about it. Just that there's an equation there is what's really important to us, because this equation allows us to relate different aspects, different geometries of this cube to the bulk mechanical properties that we can measure experimentally in the lab. Even though the cube is really tiny, it's 10 microns on a side, even though the composition may be cellular proteins like cytoskeleton, it doesn't really matter. This model is independent of what particular molecules are present. So the first problem that we ran into was that the central assumption of this whole endeavor was that the mechanics of a cellular solid, like a piece of packing foam, is dependent not on the structure, the large-scale structure, but on the individual parts of that unit cell. Right? And that's a pretty big assumption, especially when we think about embryos. We think about the embryo going from an early stage to a later stage. This is a cross-section showing us these germ layers. These are the mesoderm in here, and the ectoderm, and the endoderm. This is at the early time point, and this is later. Just as the central nervous system is about to form up here, I like to look at this, and I just like, oh my God, it looks like they're going like, bam! These pieces of tissue physically look like they have mechanical properties, like structures. You would build a bridge like this instead of like the top one, right? This would be a bad bridge. This would be a great bridge. So look at that and we think, okay, but if the embryo has a bridge built inside of it, then it's not, its mechanical properties are the structural elements of the bridge, not the bits and pieces of the cells. And so we thought, how do we test that? How do we test our initial assumption? How could we prove it wrong? Well, what we decided to do was we followed some experiments by these ancient, old embryologists that took pieces of the embryo out. As I was just telling you, you could take a piece of embryo out. You can dissociate those cells into, those tissues into individual cells and just recombine them into another tissue. So it's kind of like a blender experiment. You take something apart, you blend it, you put it back together, and amazingly, in some cases, it reforms the original structure back for you. And so we thought, well, we can't make these structures as bioengineers, but we can certainly scramble them like they did. And so we took these tissues apart. We one scrambled is on the right-hand side, and a native tissue unscrambled is on the left. I think you can tell the difference between these two. This one holds together. This one does this nice thing that it would do normally if it was inside the embryo. So this is a great example. And so does this scrambling of the tissue change its modulus? So if these big structures, the bridge-like structures were important, we just dissociated them and repacked it together in a totally mindless way. And we wanted to know whether this piece of tissue had the same mechanical properties as this piece of tissue. And so we had to answer a couple of questions first. We had to show that here's the cells are alive. They assemble different aspects of the biology are still working. And so cells are alive, yes. Do they programs of differentiation, are they doing what they would do if they were still in the embryo? And the answer is yes, they do. And then the last one was, do they restore the structure? Do they build this bridge all over again? And no, they don't. They actually don't form big structures, and they can't undergo morphogenesis, but they can make that block of tissue. And so doing the experiment, again, this was a great example of disproving your hypothesis. We found that the tissues didn't change mechanical properties, whether they were scrambled or whether you had that native architecture. This is kind of thinking about taking a bridge in Pittsburgh and taking all the pieces of it apart and then throwing it back into the river, and it still works as a bridge. All right, I don't think it works like that. I live near the Fern Hollow Bridge, and it didn't work like that. Okay. So we then went through our hypotheses individually, case by case. We tested cell size, didn't have an effect. We went and tested cell thickness, the wall thickness, and didn't have an effect. These have effects, but they weren't enough to account for the big changes that we saw. And lastly, we looked at the composition and density of the cells themselves. And this is where we found a signal that resisted. We could not disprove this, let's just say that. And so this is what we found, was that the mechanics of this big block of tissue that's maybe a millimeter across is not due to these structures at these sort of scales, but is rather due to the those molecular composition of the cell at its surface, this cytoskeleton of the cell. Okay, so I'm wrapping up. So I think I've told you how wonderful frogs are, gave you a bit of an insight into how we think as scientists about our experiments. And I think, and I love pop culture. I just gobble it up, So, we have this rich toolkit, and you can think about this as the utility belt on Batman. I like Batman because he doesn't have specific superpowers. He has really good funding from the federal government. And so I think we can understand how tissues are shaped, and as well as this mechanobiology part, and that many of our tools are useful for studies with human organoids. And so for the next 50 years or so, I think we're going to be, we're ethically constrained not to study human development directly. We cannot work with human embryos. What we can do is we can study how organs are formed in something that's called an organoid, taking these iPSCs, induced pluripotent stem cells, and making human organs or human organ-like structures. And those structures are often at the same scale and are the same robustness as the frog embryo that we study. And so these tools to study frogs can be extended to study human disease, and I think lead to some developments in regeneration and cancer. And the future is promising for this model system, the African clawed frog. We can do things called single-cell transcriptomics, the very latest, hottest techniques for looking at the genotype of individual cells, what's inside of individual cells. We can relate the cells and the genes computationally. And then I think we can build more on this sort of understanding about the biomechanics and mechanobiology of early development. And so just to bring this back and thank again the folks in my lab now and the folks who did this work back in the day. And then lastly, I really have one more slide. I really have to give the most important thanks to my wife and daughter, my mother for schlepping me off to museums such as this one from when I was just a wee tadpole. But you know, it takes a family and people who tolerate you and who travel with you and join you on your adventures to really make things worthwhile. So thank you very much.
Lance, thank you so much for that fascinating presentation. Folks, we're going to go ahead and open it up to questions. A reminder for our online audience, go ahead and use the Q&A function in there, and I will be feeding those questions to Lance. And Sammy will be running our physical microphone here in the theater. Please wait for the microphone to come to you and then speak into it.
Well, first let me say thank you for an absolutely wonderful, both charming and informative lecture. I enjoyed it. So, being that frogs is a biologist's best friend, I won't ask you if you have a dog, but I would like to know if there happened to be a particular African claw-toed frog that informed your research, or several of them that informed your research, and that you feel particularly, what can I say, affectionate about. Thank you.
Well, the secret is that we have a colony of frogs. We have over 150 female frogs and about 20 male frogs at any one time. So I think the rule in the lab is not to name the frogs, although some frogs are notable and you kind of remember them, but mainly from the quality of their eggs. So, but we don't name them. Yeah, I haven't kept frogs since I was a postdoc. We're obligated not to take them out of the lab and not to let them grow up until they are conscious of pain and suffering. So we have a very strict what's called an IACUC protocol, which is an animal protection, animal welfare protocol that controls how we interact with the animals and how we deal with them. The University of Pittsburgh's Division of Laboratory Animal Research maintains our frog colony. We go in, we can, as I say, here's my t-shirt says there's more than one way to squeeze a frog. We go in, we obtain eggs, and then we come back out. Yeah, though I do have a favorite frog. It is Kermit. It is Kermit. In fact, I just saw the Jim Henson documentary last night, and I was like, oh my gosh, that's where Kermit was born. So, yeah.
We've got a couple online. Okay, so I love that that was the first question, because our first question from our online audience is, what is your favorite frog-themed trinket you've received? People really want to know about your relationship to the frogs.
So, yeah, so relationship to frog trinkets. The truth is that this is somehow, you know, in a Buddhist kind of way, preordained. My grandfather collected frog trinkets. And he had frog spitters, like in the garden. He had so many. I mean, it was like, what does the family do? And it, you know, like hundreds of frog pieces of things. So I try not to collect anything like that. But I do have a frog from my grandfather's collection that I have out in my garden, and so that's a very special frog. Yeah.
Okay, we've got another one here online. Virginia asks, how do cells communicate so that morphogenesis can occur?
That is such a deep question. So cells communicate to each other through like every possible way. They communicate with electrical signals. They communicate with chemical signals. One cell secretes a chemical and another cell receives that signal. Cells can lay down a matrix and other cells can follow it later. It's like building a city and then the next generation of migrants come in and occupy it. And so everything you can imagine. And so the last 30 years or so has been where we've revolutionized our understanding of cell communication. We know all of the path. I think I'm pretty confident that we know all of the path, and I'll get really, that we know all of the signaling systems that cells use to communicate with one another, but we don't know much about how they communicate mechanically. So like a cell would push another cell, and that can trigger a response by itself. So we're working all of those out, and I think that's where like the bread and butter of developmental biology is. Yeah.
Thank you.
Hi. So you've obviously focused on the frogs there, but are there other scientists who've focused like on the mice or things like that? And do you interact with them to say, hey, we just found out this, does that actually work in mice or something like that?
Absolutely, yeah. So the question is, do we work on, how do we interact with people who work on other model systems of development? And so, absolutely. So there are things that we can do in frog that they cannot do in mouse. And so my dream, I've always thought about like, what's the dream organism? And the dream organism for me is a frog that's an egg that's about this big. Make things a lot easier. But like working on mice is like an order of magnitude smaller for us. But we have suggestions for them. They provide us some data. We have collaborations now with people that work on mouse neurulation and people that work on fruit fly, a process that looks like neurulation. We've had these interactions throughout my entire career. I began by working on sea urchins, and working on sea urchins, I wanted to work on something after that that was more vertebrate, but not fuzzy enough, not that fuzzy. But mouse is certainly a system that we strive to translate what we've learned into mouse, because mouse is the stepping stone to human development.
Hello, Lance.
Hi, Susan.
Everything that you've spoken about is brilliant and American and from the University of Pittsburgh. Do other countries do it the same way you're doing it?
So I represent a small part of developmental biology, and that's the part that works on the mechanics, the physical mechanics. And so, from when I first started, there were very few people doing this work. But now I go to meetings and there's lots of people in Europe and a lot of people in the United States, and they're taking this idea of physics, and can we understand the physics of the embryo? And I think absolutely, yes. Europeans are doing a great job of it. The US is doing a great job. Japan, South America. The science is global. And in fact, I'm a co-organizer of a Zoom meeting that's an international Zoom symposium. This is one of the best things that arose from the pandemic, is that scientists could start to use Zoom. They use these communications tools now, like at the drop of a hat. Somebody sends you an email and says, hey, do you have a few minutes? And you're just like, I'm there. So we have these communications across the world and it's just amazing to be able to do that. Yeah. Follow-up question from Susan.
There was Dolly the sheep.
Yes.
Who's next?
Well, I mean, it's illegal to do humans, to clone humans. I think in many countries of the world that's illegal. It's unethical to do. Some of the operations that you need to do to make those individuals are very inefficient. The frogs that John Gurdon worked with, he had to do thousands and thousands and thousands of eggs before he came up with these 20-some clones. Dolly the sheep, same thing. A number of failures is very high and exceeds what you would want for any kind of human experimentation, right? So that's a downside, yeah. So that approach, somatic nuclear transfer, is still being used in for some model systems, but I think it's not going to be used for too much longer.
We've got another online,
All right.
online question here. How do you know when a frog embryo feels pain?
So this is all often up for debate, and I mean, you can write surveys and they just don't fill them out, right? But there are ways to design experiments, and again, they have to be experiments that are designed from like a strong inference perspective. Can you disprove them? And that's where the difficulty lies. You can collect evidence that a tadpole of a certain stage will be averse to pain, but you can't show that they feel it as pain. It could be avoidance behaviors, and so by and large we err on the side of conservativeness, right? So we euthanize our embryos if they reach a stage that we think that they could start to feel pain, and we dispose of them. So we have to terminate them before they get to the point where they could feel pain, and that's the nature of our animal welfare approaches in the lab. So yeah, and the same is true for adults. If adult frogs are in pain or if they're injured, we are obligated to euthanize them. So I don't feel bad about that. I feel bad that, that maybe we can make it, we can make sure that the frogs don't have those conditions, and we work judiciously to try to prevent that. But I think that's what goes hand in hand with working with animals. We would never be able to get this kind of information if we didn't use animals in some way.
So since you mentioned in the slide about orphan black, if one had no ethics and was willing to go down that path, scientifically, was their whole concept for that somewhat feasible if you had the controversy avoidance mechanism in there to do it, and you had the government backing and the secrecy and all that?
That's a tough question. Yeah, I think it makes great television, right? It's entertaining, and we imagine what ifs. That's the role of science fiction, is to ask the question, what if this was possible? And I think I'm a great fan of science fiction, and I think we ask that question about individual identity and our sense of self, and there was huge debate early on about like all these embryos, if you take a nucleus out of one, or you clone them, or you make them, how is the individual formed, the consciousness of that individual? If they have two individuals, you make a twin. Are they the same? Do they share the same consciousness? All these kind of questions. It's way above my pay grade, I like to work on tractable questions that can be falsified, right? So, yeah, I don't know. I don't know how to answer that. Yeah.
So I noticed in the video you were playing of the embryo forming, how it kind of seemed to fold.
Yes.
And all of the animals that you mentioned that you use commonly in the lab all have bilateral symmetry, but you
Right.
just mentioned urchins. So in some of those other animal development, do you see other patterns kind of come out in that embryonic formation?
Yeah, so urchins is an interesting one. And so urchins, the early urchin development is bilateral symmetry, bilateral. And then only when it undergoes metamorphosis does it develop this, this multifold symmetry. And even inside the animal, there is still some, some little bit of this, this twofold symmetry, right, still present. There's a fantastic work going on by a colleague of mine, Chris Lowe at Stanford, who's working on just how is it that organisms like sea urchins have evolved. How did this pentameric symmetry evolve? And he's got some really amazing sort of thoughts on it. One is like that they're all head, and that somehow they like invert the head. He demonstrates it with his hands in a way that I'm uncomfortable with. So, but these are great questions. I mean, that's a great question. It's part of where our work kind of gets at like what is the possible way of doing this kind of manipulation, this kind of morphogenesis. How is morphogenesis possible under those circumstances? Is bilateral symmetry always going to happen? And no, that's not true. There's plenty of organisms that are not bilaterally symmetric.
I actually have a question, if that's okay. I'm over here. Hi.
Oh, yeah.
Hello. Never asked a question before. So here at the Science Center, we actually have a show called Frankenscience, where we talk about Frankenstein and a little bit about cloning, and we talk about Dolly. And I like what you mentioned on your slide about how, you know, the cells, when you like open them, you can put, in the 50s, you were like, they were crazy, doing all this crazy stuff, put in little pieces and other pieces. So where, for you, in your opinion, do you think the line is drawn between, you know, what's needed for research and quote unquote mad science?
Oh boy, yeah. What's the difference between the science in the lab and the science that's published, and the mad science? So one of the things that this principle of strong inference it seems really so rigorous and like so controlling, but there's been sort of a revision of that program. There's a guy named Douglas Fudge who's written like, how do you take strong inference and make it better? And part of that is by having a phase. How do you discover these hypotheses? How do you actually find a new hypothesis? And so he proposes this sort of phase of curiosity-driven research. And I like that. I like curiosity-driven research. I encourage my students to try things out, to throw a bunch of stuff against the wall and see what works. And we make discoveries like that. That's serendipity, what's surprising versus what is not surprising. And so I think we look for that in our research. And I think it's important to inspire curiosity and creativity in the lab. It's really important. I mean, and yeah, I think that's like mad science can look like that. I had sort of like wagers when I go teach a course. I was like, oh, this experiment where you can take a piece of the embryo and put it somewhere else and it makes a twin. I was like, well, let's try putting two wholly formed embryos together and see how they work. And you can do it, and it works. It's just, it's not something, Frankenstein is kind of what we can do. We can take pieces and put them together. There was a demonstration once where a colleague of mine was demonstrating a new kind of surgical tool that would cut through tissues very quickly. And so he made what was called a stretch limo tadpole by making, putting extra pieces in the middle. And so that's a possibility. That's, what do you learn from that? You learn that the healing mechanism is awesome, but you also learn that these segments can be don't really depend that much on their exact position. They can reintegrate and they can form properly. And so you learn things from those kind of experiments, yeah.
Thank you.
Yeah.
My follow-up question to Sammy's amazing question about mad science and yours about how we can often learn things kind of, even if that was not the thing we set out to learn with these experiments. So I've read elsewhere about scientists, and I'm not going to remember, exactly who or where, but there's been some number of experiments with removing bioluminescent cells and placing them into animals that do not normally bioluminesce. So a very important question is, when will I have a glow-in-the-dark cat? Because I really need that.
Actually, there is already a glow-in-the-dark cat.
Well, can I have it?
I think you have to talk to them.
Do you have any ins?
No, I don't do the cats, so, frogs. I could point you to where you could get some fluorescent frogs. And there on the market, there's fluorescent fish. There's something called glowfish, which are transgenically constructed fish with green fluorescent protein or red fluorescent protein placed into their genomes. And so I have not yet seen body mod people utilize this. It requires fluorescent UV light to make possible. That's not a good thing. So, but yeah, I think that's absolutely, yeah.
Thank you.
Yeah.
Do we have any more questions here in our on-site audience? Oh, looks like we have one right here. I'll hand my mic.
You said earlier, or actually it was on the board there, that you come up with a hypothesis and then you prove a bunch of them aren't true, and if you can't prove it's not true, that makes it true. That sounds completely opposite as I understand science. Can you say anything more about that?
Yeah, so that is. It does sound quite counterintuitive that you try to disprove things. And that's the sort of the power of this strong inference approach.
Well, you disprove, but you also prove.
What's left?
Because that's what's bothering me. I see science as proving things.
See, you can never do that. I think that's totally, you're misled. So what you can do with science is disprove things. The proof is actually hard to come by. And it's hypotheses that stand the test of time that get turned into law, right? In science, that's sort of the difference between hypotheses that have been resistant to falsification and laws. That's really where the heavy decision-making comes. But when, like Newton's laws came into play, they were formulated to account for a whole series of observations. Tycho Brahe sat outside, tracking, the star movements, tracking planetary movements, and through lots and lots and lots of observations, they formulated a model and then tested that model. And we call now a law. Now we think that that was proven by someone, but it really resisted falsification again and again and again. And so I'm confident with calling gravity, a law that's no longer a hypothesis. But in modern biology, I think we're still in that kind of hypothesis phase where some things sound really good and once they're subjected to these kind of tests, they fall to the side. So things that sound good, I'm a cynic. I don't trust anything that sounds good. I want to see it actively withstand a rigorous test before I'm confident to use that. Yeah. So that's like my union card, right?
It's just a different way of looking at it in my mind.
Yeah.
In science, there's nothing in science that is absolutely true. It can be eventually proven to be false, or get more information that makes it modified.
That's what we live for. I mean, I'm so glad when I disprove my own hypotheses. I feel much better when I disprove it, or someone in my lab disproves it, rather than a colleague in Europe disproves it. Boy, that's a bad day, right? So, but I think that's what we strive for. We strive to disprove our hypotheses. And in fact, when I was a postdoc, I spent years characterizing a structure in the embryo. Beautiful. It had all the hallmarks of something that would be mechanical. And the first couple of years in my own lab, we went and knocked out that structure, and it had no effect on the mechanics. And so it was both a wonderful day in the lab, but also a sad day, because it was my hypothesis that died. So, but I'm happy with that. I could get up in the morning and have a good day. So I live for that. Yeah. Right.
Thank you.
Thanks.
Thank you. Okay, it looks like we've taken care of all of the questions we have this evening. So let's just give Lance another big round of applause. Thank you so much. Man, I'm loving this love for you. And I'm just going to remind everybody else that please come back again and join us or tune in next month for our next Cafe Sci. And don't forget to stick around and check out the teeny weeny frog embryos that Lance brought in his bag. I saw them. They're very cool. So come down here and we'll look at some tiny frogs together.
Yeah.
Okay. Thanks, y'all. Have a great night.
Thank you.
Please don't forget to take your trash with you.